Welcome back. Let's jump right into today's explainer because we're looking at something genuinely revolutionary in medical research. If you track how new medicines get to market, you're already familiar with the usual pipeline, right? Phase 1, 2, and 3 clinical trials. But what if we could drastically speed up that whole development process before those traditional trials even start? Well, that's exactly what we're going to demystify today. We're going to explore phase 0 and the incredible power of the microdose. To really grasp phase 0, we've got to wrap our heads around the sheer scale, or actually, the lack of scale of a microdose. We are talking about less than a fraction of 1% of a typical active dose. Imagine taking a standard pill, slicing it into more than 100 microscopic pieces, and just taking one of those tiny dust-like fragments. It's an impossibly small amount, but believe it or not, it yields a massive amount of biological data for scientists. So let's kick things off with Section 1. What exactly is Phase 0? Phase 0 is actually a highly specific step. It's the study of a new drug in microdoses to derive early pharmacokinetic, or PK, information in humans before we ever hit Phase 1. Instead of just crossing our fingers and relying solely on animal models, Phase 0 safely gathers this early biological data on how a drug is absorbed and metabolized. And it does this using amounts way too small to trigger any real bodily response. Now, a microdose isn't just some arbitrary small amount. It has a very rigid scientific definition. By rule, it has to be less than 1/100th of the dose calculated to actually produce a pharmacological effect. Plus, there's a hard ceiling. The absolute maximum dose is capped at just 100 micrograms. To put that into perspective for you, a single grain of table salt weighs around 50 to 60 micrograms. So we're talking about a maximum dose that weighs about the same as 2 grains of salt. The reason for this extreme limitation? Pure safety. It completely prevents any major pharmacological effects from happening in the human volunteer, while still letting highly sensitive lab instruments track exactly where those 2 grains of salt go inside the body.
Which brings us to Section 2, the objectives of microdosing.
Obviously, the primary goal here is getting that preliminary PK data. But a quick side note, this doesn't mean we just skip animal testing entirely. Preclinical data, specifically a subacute toxicity study in one animal species using 2 different routes, is still a strict prerequisite. But once that baseline is met, researchers can safely evaluate both pharmacodynamic and pharmacokinetic properties in humans really early on. And the ultimate objective? To drastically accelerate the entire drug development pipeline.
And that acceleration directly translates to finding the best candidate faster. Think about it like this. Say a pharmaceutical company has 10 potential chemical compounds that might cure a specific disease. Putting all 10 of those through full phase 1, 2, and 3 trials, that would take decades. It would cost billions of dollars. But by using phase 0 to get that early human bioavailability data, researchers can quickly see, which of those 10 compounds actually survive the human digestive tract, or which ones just get immediately destroyed by the liver. It helps weed out the duds early and lets us select only the absolute most promising candidates for the real clinical trials. It's literally the ultimate filtering mechanism.
All right, let's look at the flip side in Section 3, weighing pros and cons.
When we weigh this whole approach, the operational benefits are pretty obvious. You have a vastly lower chance of adverse events, much shorter trial durations, way fewer human volunteers needed, and ultimately significantly reduced costs. But scientifically speaking, the limitations are actually pretty severe. For one, it relies entirely on PK parameters, which means it tells us absolutely nothing about whether the drug is safe or effective at a real dose. Second, it completely misses agents that behave differently at a micro level compared to a full dose. And third, sure, the overall trial might save money, but the specific lab requirements, they depend on incredibly expensive bioavailability and bioequivalence labs. I mean, the highly sensitive machines needed to track our 2 grains of salt definitely aren't cheap to run. Let's drill down into that first major limitation, what we can call the efficacy catch. It is absolutely vital to remember what phase 0 does not do. It does not prove safety, and it does not prove efficacy. Yes, we learn exactly how the body absorbs, moves, and excretes the drug, but this phase completely skips over whether the drug actually works to cure the disease, or if it might cause terrible side effects when given at full therapeutic doses. A drug could look absolutely perfect in phase 0, but completely fail to help a patient later on in phase 2. And then we have this tricky technical hurdle known as the nonlinear problem. See, some chemicals behave linearly, meaning a small dose acts exactly like a large dose, just on a smaller scale. Simple enough, right? But many drugs have what's called nonlinear pharmacokinetics.
That means the drug behaves drastically differently when you scale it up to a full dose. Maybe at a microdose, the liver processes it no problem. But at a full dose, the liver gets overwhelmed and the drug builds up to toxic levels. So for agents with nonlinear PK, microdosing is practically useless because the data you pull from the microdose doesn't accurately predict what will happen with the full therapeutic dose. It's a massive blind spot in the whole phase 0 strategy. All of this leaves us with a final, really critical question to ponder. Do the massive cost and time savings of running a phase 0 trial actually outweigh the risks of those expensive lab requirements and the blind spots caused by nonlinear PK agents? It's a delicate balance, and it's one that medical researchers have to evaluate every single time they develop a new potential cure. Using a microdose to cheat time is brilliant, but for sure only if you truly understand the limits of the data you're collecting. I hope this explainer helped clarify the fascinating world of phase 0 and how the absolute smallest doses are shaping the future of medicine. Thank you so much for joining me today, and please like, share, and subscribe to our channel.